Okay. Okay. Vice Provost, colleagues, ladies and gentlemen, you're all very welcome. Thank you for coming this evening. My name is Brian O'Connell. I'm Dean of the Faculty of Health Sciences, who's hosting this inaugural lecture this evening. The inaugural lecture is a tradition that's afforded to full professors in Trinity College, and so it gives me great pleasure this evening to introduce Professor Maeve Lowery, who will shortly present her lecture, The Arc of Progress in Precision Cancer Medicine, How Far Have We Come and Where Do We Have to Go? So Professor Lowery is Professor of Translational Cancer Medicine here in Trinity College, Consultant Medical Oncologist in St. James's Hospital, and Academic Director of the Trinity St. James's Cancer Institute. Maeve describes herself as a clinician first and foremost, and this has guided her career trajectory, I think, right from the outset. As a student and junior doctor in the Mater Hospital, Maeve was greatly influenced by Professor Des Carney, and her interest in oncology began. After a rotation in the Mayo Clinic in Scottsdale, a year in St. James's, Maeve headed to Memorial Sloan Kettering Cancer Center in New York, where she completed her training in gastrointestinal and pancreatic cancer and met, among others, Professor Eileen O'Reilly, who was another inspiration and role model. Professor O'Reilly was equally impressed by Maeve and said, Professor Lowery is an outstanding colleague, incredibly smart with vision, thoughtfulness, and productivity, and highly accomplished in her field. She is to be congratulated on her highly deserved academic success. A fellowship followed for Maeve and then appointment to the faculty in Memorial Sloan Kettering. Maeve and her family were settling into a comfortable life in New York and starting a family. The attractions of being able to provide advanced clinical care and to engage in cutting-edge research are obvious. But as often happens, there's a call from home, and in this case, a chance to take up a unique academic oncology post in Ireland as Professor of Translational Cancer Medicine in 2018. Maeve was impressed by the ambition and the vision to establish the Cancer Institute and the opportunity to really improve cancer care for patients. Paul, I'm sure, turned on all his charms, powers of persuasion. But in treating a particularly difficult and destructive disease, Maeve decided that as an academic, there may be some time and space to address the challenging problems, and as she put it, create the bandwidth for some hope. She still describes herself as a clinician at heart, but there is much more to it than that. Maeve has been a key part of the development of the Trinity St. James's Cancer Institute from the outset, from lead on clinical cancer trials, the development group, OECI accreditation, and as academic director since 2021. As her colleague told me, her track record of achievement is matched only by her determination, persistence, and enthusiasm, with all of this underpinned by a clear long-term visionary approach, which will continue to serve Trinity and St. James's for many years to come. All the while, Maeve has been leading with her own pioneering research, of which we will hear more in a moment, attracting funding from all the big agencies and contributing to a very long list of national and international advisory boards, coordinating groups, and advocacy organizations. Co-workers told me that Maeve is a great collaborative and supportive colleague who shows positive leadership in every role she takes on. She always has a positive can-do attitude in tackling all problems. She's a true leader with a passion for making personalized medicine a reality for all patients. Maeve is known for balancing all of these many commitments and her family, yet finding time to provide mentoring support and friendship to those around her. As one person who knows Maeve well noted, she's a true exemplar of the successful academic translational clinical researcher, a role to which many aspire, but fewer perhaps truly achieve. Professor Maeve Lowery, we welcome you and look forward to hearing your thoughts. I invite you to give your lecture.
Thank you. Thank you, Dean, for that introduction. That was it's wonderful, and I'm really happy to be here today to give my inaugural lecture. I'm not a new colleague really to any of you, am I? Because I've been here for a few years. But because COVID came so soon after I arrived, it's just really nice to appreciate the chance to gather together and to have a lecture together. And I thank you all for coming tonight. So I've chosen to focus on precision cancer medicine or individualized cancer treatment for my lecture, where we've come from and where are we going. And it's about 20 years since I was a medical student in my first rotation in medical oncology in the Mater Hospital with Des Carney. That's when I first began seeing patients with cancer. And I expect that I'll be seeing patients with cancer on this campus for about another 20 years or so, unless I bump up my pension contributions significantly. So it seemed like a good point in my career to look back at the progress that's been made in cancer treatment over the last 20 years and look to a patient that targets fast-growing cells. You're not just going to target the cancerous cells. You're also going to damage the patient's normal cells, which are also replicating. So chemotherapy causes decreased turnover in cells in the hair follicles, so our patients lose their hair. In the bone marrow, so patients become anemic or have low blood cells, white blood cells, that put them at risk of potentially severe infections. It damages the lining of the stomach that makes them get nauseous and makes them vomit. And it causes a reduction in the lining of the colon, which gives them diarrhea or sometimes constipation. So the problem with chemotherapy is that in damaging the cancerous cells, we have to also damage the patient's normal cells, impede the replication of those cells that the patient requires to remain well and healthy. And that leads to side effects that causes our patients significant suffering and can lead to severe side effects. And sometimes, in the worst scenarios, leads to death of the patient from the treatment, from being worse than the disease. So how do we do better than that? Well, a more nuanced approach is to figure out why the cancer cells are growing fast and go after that. So rather than just target the fact that the cells replicate quickly, we can identify the mechanisms by which they do so. And by targeting upstream, identify those mechanisms, block them with rational treatment strategies, we can have better effect against the cancer cells and less of an effect on our patients. So when I started in the Mater Hospital in oncology as a house officer, one of the first patients I cared for was a man who wasn't much older than me. He was in his early 30s. And he had been traveling and backpacking around Australia, and he had developed a malignant melanoma on his back. And he traveled and he enjoyed life and he returned to Ireland. And then he developed some pain in his abdomen and came into the Mater A&E. And it turned out that the melanoma had spread to his liver and to his lungs and to his brain. And in the early 2000s, at that time, the standard of care treatment for malignant melanoma was a chemotherapy called dacarbazine, which the oncologists here will remember. We don't use it much anymore. And this patient had a drive to live. He tried dacarbazine, he tried as much chemotherapy as he could, but melanoma is a very chemorefractory disease. And that man passed away just a month after he got married to his then girlfriend in his early 30s. But at the same time, in this almost the same year, a discovery was made regarding malignant melanoma. And that was the identification of the importance of the BRAF mutation in malignant melanoma. And it turns out that almost half of cases of malignant melanoma, the tumor harbors a mutation in this BRAF gene. And mutations in that gene cause acceleration of signaling through a particular pathway that accelerates the growth and differentiation of the melanoma cells. So the melanoma cells become very dependent upon this particular mutation. And so following on from that, were developed inhibitors of this mutant protein, which blocked the growth signaling that came through those cells and blocked the signaling that helped growth and differentiation of the melanoma cells. So as I was moved to Sloan Kettering in 2009, clinical trials of this agent were just completing. Single tablet taken once a day, It just targeted the mutant BRAF protein, so it spared the patient's other cells and caused minimal side effects. And so this, for the hope there are no radiologists here, oh, there's one I see. This is a PET scan, and I'm not a radiologist, but essentially what you're looking at here, on your right-hand side, that picture, is a baseline PET scan, an image of a patient taken prior to treatment. Now, PET scans really are just when you inject a labeled glucose into the patient's bloodstream. It's labeled so you can image it. And areas of the body that use a lot of glucose, that have a lot of high metabolism, those light up bright on the scan. So obviously your brain lights up bright and your heart lights up bright, but also areas of cancer light up bright because they are high metabolic areas. They use an awful lot of glucose. And so you can see in the image to your right that there is an awful lot of areas around the patient's neck, around the patient's chest, around their abdomen. The patient has melanoma throughout their body. And that's a patient who's failed multiple lines of ineffective chemotherapy. And the next image you see is at day 15, after 2 weeks of treatment with a single tablet targeted treatment, targeting the mutant BRAF protein. And even the non-doctors among you will be able to see that there's been almost complete resolution of all those areas of cancer with 2 weeks of a single tablet given once a day. And that's mind-boggling. You think of the damage we cause with chemotherapy, and then the fact that one could treat a cancer as devastating as this with such a simple, smart method really made us all hope that we're moving beyond chemo. Forget about chemotherapy. We can move ahead and do better. We target just the genes, doesn't matter where the cancer started from, and tablet therapy will lead to better outcomes for our patients. And to some extent, that's true, but in fact, it's more complex than that. I suppose to explain what targeted therapy means, and I won't spend long on genes, but just to explain what we mean before I move on, I suppose in terms of molecular targeted therapy, when we talk about that for cancer, we're talking about using information from the cancer genome to develop better treatments. So genes are just really the blueprint by which cells make proteins. So by understanding those genes, by identifying how they can change themselves, mutations that occur in the genes can cause different proteins that help the cancer cells grow and divide better. If we understand those mutations better, we can engineer drugs that block the activity of those proteins and slow the growth of the cancer. And the nice thing about these drugs is because they block a single target, they spare the patient's normal cells and they cause usually less side effects. Well, it sounds very simple and very straightforward, and the concept seems somewhat obvious, but in real life and in practice, of course, it becomes an awful lot more complicated. So I completed my fellowship in New York in 2011, and I began on faculty there in the GI oncology department. And it was a time, even though it's not that long ago, it seemed like a different era because the excitement around targeted therapy and maybe our naivety around the development and the simplistic development of it was perhaps less nuanced than it now is. So I was lucky, as Brian mentioned, to work with Eileen O'Reilly, who's one of the foremost clinical investigators in GI oncology in the world. And she allowed me to be the principal investigator on this really interesting study. And that was the first study I led on my own as an attending as a consultant in Sloan Kettering Cancer Centre. And it was a great study because this was a first in human study. This was the first time that we were trying this particular drug that blocked this particular mutation. And it's a really interesting mutation because most of the time when there are mutations in cancer causing or cancer driving genes, they either hyperstimulate a pathway that already exists or they block a pathway that already exists. But this mutation is what we call a gain of function mutation. It causes the protein made by the cell to do something that's totally different, that's not done at all in non-cancer cells. It causes it to make a protein, which is an oncometabolite, that causes the cancer cells to grow and grow and grow and grow and grow. And so this was the first time we were trying in solid tumor oncology a drug that blocked this particular protein. And so it's found very commonly in bile duct cancers, and that was the main cancer that I treated in my initial years in Sloan Kettering. And when I opened the study, it was one of the first sites to open in North America. So patients have been waiting for it to open. And the first patient I put in the study was a businessman, very, very smart man, who flew in from elsewhere in the US. He was in his 70s. He tried all treatments for his bile duct cancer. And he had had genetic testing done of his cancer when it wasn't standard of care. He'd paid for it, he'd known to get it done. He'd researched carefully the results, and he'd found the right trial for exactly his subtype of rare cancer, and had flown into New York to start the study. And it was a time of great hope, the first patient put in the study, and he was, without other good treatment options. And we started with great hope, and checked him every week, and I watched him get sicker. It didn't work. And within a few months in the study, he got even sicker, and he succumbed to his disease. And I thought, gosh, what have I opened here? I mean, this isn't great. Should I just put anyone else on it? What should I do? But I checked on my patients to see if they got the mutation too. And I had a whole load of patients who had the mutation. I wondered, maybe I should try another patient. And so the next lady who had that same mutation in her cancer cells, she was a Russian lady. She's been my patient for a couple of years. And we'd never spoken really because, she could only speak Russian, and we had this big burly interpreter that would come to help us communicate. But it was kind of challenging. And so she by the time she went on the study and decided to go on the study, she had a few symptoms from her cancer. She hadn't a lot to lose from it. She understood the risks, and she decided to go ahead and go on the study. And when she went on the study, she had some fluid on her belly, and she had some pain. And she had to choose at the time, a bit brutal in America, you can't go on hospice care while you're also getting hospital care. So she had to choose between active treatment in the trial or just supportive care alone. And she chose to go on the trial. And she came to see me just 2 weeks after she started, and the fluid had gone, and she had stopped all of her pain meds, and she had put on almost 1 kilogram of weight just in 2 weeks, just on the same drug, same disease, same mutation. It was astounding, and she became what we call an extreme responder. So she was over almost 2 years in the study. She had dramatic shrinkage of her tumors, and she had a good quality of life over that time. And the next girl who came to me to go in the study was almost my own age, young. She had purple hair. She was a skier from upstate New York. And when she started the drug, she couldn't breathe because of cancer in her lungs. And again, 3 months after starting the trial, she went on a skiing holiday with her friends. But in between all of those patients, there were others who got sicker, as though I was giving them water. So how do we square that circle? And this drug was actually FDA approved for treatment of bile duct cancer not long ago. Approved because it helped some, not because it helped all. And so it's not so simplistic a concept that we originally would have thought, whereby having a particular mutation is sufficient for response to targeting that mutation. It turns out that the mutation is required but not sufficient for responding to a targeted therapy. So it gets a bit more complex. Well, how do we deal with that? How do we move forward from that? And the answer is translational cancer science. The answer is translational medicine. Because all the patients that were treated in that study, in an incredibly generous manner, underwent research biopsies before treatment, on treatment, and even after they progressed on treatment. Imagine how generous that is, And so that allowed us to take samples of blood and of tumors, and to sequence them in the laboratory, to grow them in the laboratory, to grow them in mice, to take samples of blood and to sequence that, to look for shedding of the DNA into the blood, and to use all that information to take it back to the patient's story and try and figure out why one patient had responded, why one patient had not. And one of the biggest discoveries in that came from that Russian lady who provided us with all that tissue, and both at pre and post her treatment. And it allowed us to identify the primary mechanism of resistance to this drug. And believe it or not, in her cancer had managed over the course of the 2 years to develop another mutation in another gene. And it turns out the mutation was in the only other gene in the body that could also produce that protein, the oncometabolite that her cancer was so dependent on, IDH2. Amazing. So that was, I suppose, an illustration of the generosity of the patient, that she was so generous with her time and with her efforts to provide tissue that it actually not just helped us learn from her clinical course, but helped us correlate her clinical course with what we found in the lab, and then identify changes that help us to bring forward the drug to other patients in a more meaningful, in a more targeted way, and overcome that kind of resistance. So molecular targeted treatment is here to stay, but how are we moving forward with this? What does the next decades hold in terms of that? Well, we have, I suppose, a couple of ways we can move forward beyond the simplistic targeting that we've shown you. And the main part of that is really how we look at cancer genes. So typical way we look at cancer genes is to look at maybe one gene we suspect to be altered. or you can look at 2. Nowadays, you probably look at maybe 50 at the same time, even maybe up to 500 at the same time. But we also have the capability in research, if not just in clinical care, to look at the entire region of the genome that codes for proteins, exome sequencing. And we also can, with even small pieces of tissue, look at the whole genome, including all the dark matter that doesn't really code for anything. And it turns out that sequencing the cancer is actually kind of the easy bit, but interpreting all of that, taking all that information we get from the sequencing and bring it down to a patient level to make decisions, well, that's actually the really, really hard bit. So this is work done by Emily Harold. She's currently at Sloan Kettering. She's an MD student. And she took, at St. James's Hospital, patients who were with the rare subtype of gastric cancer, and also patients who were early relapsers or long-term survivors after their surgery, and did whole exome sequencing with Stephen Finn here, and identified in particular mutations that correlated with survival in those patients, particularly P53, and also ones that were identified with worse prognosis. And then I suppose perspective moving forward, more interesting is, well, it's easy to take samples from a biobank and do that, but can we do it with a real living patient who's going to come back to clinic, and can we impact and make changes that might help their treatment course in real time? And Linda Corrigan, who's another MD student, just started as a consultant oncologist in Tala, had led on this program, which is an SFI funded industry co-fund through Precision Oncology Ireland. And this is a collaboration academic industry with British Columbia Cancer Centre. And what we plan to do on this study is to take patients who have gastrointestinal cancer, that is without other good treatment options, to take research biopsies of their tumor tissue and also their blood, to do whole genome transcriptome sequencing, so read every letter of DNA and RNA, and then analyze that and figure out within that patient's cancer, at that point in time when they need a new treatment option, where are the vulnerabilities? What can we do to target it better? What are the good treatment options we might have going forward? And then, importantly, to take that information with our colleagues in British Columbia and meet with the tumor board, with geneticists, bioinformaticians, oncologists, surgeons, drug development specialists, identify what the best treatment for those patients might be, and develop a personalized treatment plan, and then bring that back to the clinic again and develop a plan for the patient, be that a clinical trial, chemotherapy, immunotherapy, or a targeted therapy. So that's precision oncology. That's ambitious because normally we do this in research, not with patients who are alive and waiting for the results. But that's really the test we have to withstand, isn't it? There's no point doing it, only looking backwards. So this is more ambitious, but to see, can we actually implement change in a patient's treatment plan, utilizing all that information? And more importantly, I suppose, the bigger picture looking forward, the future in precision oncology lies in collaboration. And we've been lucky to join these 2 large EU consortia led by our Nordic colleagues in Norway and in the Netherlands, Personalized Cancer Medicine for EU, the Prime Rose Consortium. What these are doing are looking on a national basis across the EU, and we develop better infrastructure for all those pieces of the puzzle that I showed you. The sequencing part, the interpretation part, the access to drugs part, and then also the specialist meetings of the people who need to work together to identify the correct treatment course for the patient, to bring it back right to a patient level. So we talked a bit about the genome, and I suppose that is so important, and we talk about targeted therapy for cancer. That's kind of really what we tend to focus on. But there are other components of tumors that vary a lot by cancer type and in between individuals, and it's hard to understand that just by looking only at the genome. So it is very important that we model patients' cancer in the laboratory, and a big drive forward in precision oncology development is to develop accurate patient-derived models that we can study in the lab to help develop better treatments to bring back to the patient then in the clinic. And when I worked in Sloan Kettering Cancer Centre, I was in the Jason lab in the Rockefeller Centre, and that was a lab that specialised in transgenic models of cancer. And so I worked with a mice model of cancer where all the mice developed pancreas cancer by 6 weeks of age. And they succumbed to their disease by about 12 weeks of age. They all had mutations in BRCA, and so we were able to use that information to translate some of our findings in drug development back into the phase 1 trials and the clinical trials in Sloan Kettering of agents targeting DNA damage repair and targeting the pancreas to develop better studies and trials for our patients. But part of me inherit kind of distaste of causing a living animal to get a cancer. It also turns out that mice are different to humans, both in their tolerance to drugs, their immune systems, and the makeup of the cancers that they develop, the microenvironment of that. So more recently, it's become clear that growing cancer cells in a 3D culture that mimics its corresponding organ may be a better and more renewable model in terms of evaluating patients' cancer in the laboratory. So Oh, yeah. So Johanna Kostoof is a PhD student at a Marie Curie ITN, working with Professor Kevin Conlon, myself, and Naomi Walsh in DCU. And she's been working to establish these 3D cultures of pancreatic cancers taken directly from the surgical theatres in St. Vincent's Hospital, develop them in 3D culture. And she's working to find drug screens, better treatment options for pancreas cancer, and also share them EU-wide with the consortium to develop better treatments national-wise for pancreatic cancer. That's her there with her favourite organoid. What does the future hold? Well, you can envision maybe a future where every patient might have this kind of little avatar back in the lab, so that instead of just trying the drugs in the patient, maybe we can try them on the 3D culture in the lab before we have to bring it back to the patient. Maybe we'll actually be able to develop for every patient a model like this, test the drugs in the lab, bring it back to the patient, and hopefully improve more accurately a precision treatment for that patient. So we spoke a bit earlier about how cancer cells have all these unique properties and they differentiate themselves from the non-cancerous cells. And it follows then that the body's immune system, that can kind of recognize these differences. And even as far back as the early 1900s, there were reports of spontaneous regression of cancers. Well, why did cancer spontaneously regress? It's because for some reason that patient's immune system has got the capacity to recognize that cells as foreign and to kill the cancer cells. And that was kind of proof of principle that the immune system could be leveraged or used as a treatment for cancer. And then we discovered that, well, immune surveillance also is a concept that's true. And that means that the immune system is actually constantly vigilant and suppressing the development of malignancy all the time across humans. Cancers that never even develop into tumors, but are captured by the immune system at a microscopic stage. And so all these mechanisms were fairly clear, really by the mid-1900s, but moving that forward into drug treatment took an awful lot longer. And that lag was probably about 50 years until really a proper breakthrough was made in identification of checkpoints, immune checkpoints, as being crucial in immune cells. So really an important part of the immune system is this ability to recognize the difference between normal cells in the body and those that sees as foreign, like germs or cancer cells. And that allows the immune system to attack the foreign cells, but leave the normal cells alone. And it turns out that part of how the immune system does this is by using checkpoint proteins on the immune cells. The checkpoints act like switches. They need to be turned on or off to start an immune response. And cancer cells can find ways to use these checkpoints to avoid being attacked by the immune system. So medicines known as monoclonal antibodies, these kind of targeted drugs, they can target these checkpoint proteins. They're called immune checkpoint inhibitors. These have been a massive breakthrough in cancer treatments. They don't kill the cancer cells directly. They just work by helping the immune system find better ways to attack and kill the cancer cells wherever they are in the body. And so these again, brought in a huge breakthrough in terms of hope in cancer treatment. And we began for the first time using these in many cancers to see patients with metastatic disease having longer term survival. So we really found that some of these, in some patients, not all, but some, maybe were hoping to achieve perhaps even a cure in patients previously considered to have an incurable cancer. But unfortunately, it didn't really work so well in the kind of cancers I treat. And why is that? It's because we consider some cancers to be cold and some cancers to be hot. And cold cancers are the kind of ones that don't allow much immune cells into the center of the tumor. The hot ones are chock full of activated immune cells. So it makes sense that if you take the brakes off the immune system and allow the immune system to activate against the cancer cells, it's going to work an awful lot better if that tumor is packed full of immune cells than those that don't allow the immune cells in. It's a bit like you have, the army all primed and ready to attack, but if it can't get over the ramparts, it's not much good if it can't get to the tumor, right? And so a lot of our work currently is focused on trying to make a cold tumor into a hot tumor by combining things with drugs, targeted therapies, chemotherapies, and so on. And what does the future hold in that area? Well, I suppose it's really, our hematology colleagues are far ahead of us in this way. CAR T therapy or cell-based therapy is likely to be the future of immuno-oncology for at least solid tumor and hematologic malignancies, I would say, in the next decades. And our colleagues here in James are already doing studies and standard of care treatment with CAR T. CAR T is really, in a very simplistic way to say it, and sorry Elizabeth, very simplistic. It works really just by taking the T cells out of your body, out of your blood, genetically modifying them in a lab so they're better at finding and killing cancer cells, and then putting them back into your blood to fight cancer. It doesn't work quite so well for solid tumor cancers. And so a newer therapy, still in development, but promising for solid tumor oncology, is tumor infiltrating lymphocyte therapy. And this is where we surgically remove a piece of tumor, dissect it to isolate the lymphocytes, expand it in the lab, and infuse it back into the patient. And these lymphocytes actively destroy the cancer cells, and then they leave the healthy cells relatively unharmed. So we're using cell-based therapy, really, instead of drugs, the patient's own immune system to empower against the cancer itself. And here on this campus, we have a consortium funded by Enterprise Ireland Disruptive Technology Innovation Fund, which brings together academic and industry collaboration to try and develop this type of therapy on this campus. And this is led with Aidan Long and Jacinta O'Sullivan. And it's really exciting because it brings together the expertise in immuno-oncology within the laboratory with also manufacturing capacity in the hospital to try and see if we can actually both identify, refine, select and deliver the therapy right here on a campus with co-located hospital and university input. So lots of hope, and I suppose there is a lot of optimism ahead in terms of cancer treatment. For certain cancers we've lagged behind, and in particular that is liver cancer, stomach cancer, esophageal cancer. Those are the cancers we've made least impact in over the last couple of decades. And you can see that for a lot of those cancers, the incidence and mortality remain almost the same. And a little bit more worrying again, in particularly gastrointestinal cancers, is the identification that the incidence of these cancers is rising a lot in patients under the age of 50. We don't understand why that is. It's really not clear. So these are patients who are young, fit, well, healthy. They don't have the typical risk factors for cancer that our older patients perhaps might have. But there's been a fairly dramatic rise, particularly in colon cancer, pancreatic cancer, bile duct cancer, stomach and esophageal cancers over the last decade in particular. And we can see that really rapidly rising curve, the incidence over time growing. So why is that? We don't know. Again, we can use translational science to try and figure that out. Oana Diak has just started an ICAT PhD, very bright PhD student, also medical oncologist, and her focus is going to be in her PhD on looking at the immunogenetic modifications or things that might happen within those young patients to cause cancer at such a young age and improve treatments. So this is sad in a bit, right? But it's a talk about cancer, so it kind of has to be a bit sad. Otherwise, I'm not being very genuine, right? And the reason I kind of like this poem is the last line of it. I know I do not approve, and I am not resigned. And many people here will have lost someone you love to cancer, or maybe had cancer, maybe have cancer, and many of you treat people with cancer. And so the kind of cancers I treat, a lot of my patients, they succumb to their disease fairly quickly. So on the research side, we have all this hope and innovation and optimism. And then in clinic, we still rely a lot on chemotherapy. So I show you all these new things, but in clinic, I still prescribe an awful lot of chemo. And I find myself saying an awful lot of time to patients, oh, I wish I'd better. I'm sorry, I wish I had something better. And by making that decision, will I get more toxic chemotherapy or more side effects, or will I stop treatment? It would be nice to offer something better. So I'm brought kind of firmly back to reality when I step onto our chemotherapy day ward and when I do my outpatient clinic, as many of you also in James's are. And so I spent a lot of time, as the Dean mentioned, since I arrived here, working on the Cancer Institute, TSJCI, our Trinity James's Cancer Institute. And why have I done that? Because there's clearly a lot of interesting things you could do without this. Lots of fun projects in the lab, lots of good research, clinical research, laboratory-based research. And I've done that because it matters. And it matters to patients. Because, at the end of the day, cancer research and successful cancer research is not measured by publications really or by grants. It's measured by the impact you have on patients' outcomes for patients with cancer. And so the TSJCI, the Cancer Institute, looks to integrate those two worlds, the aspirational, forward-looking, problem-solving academic world with real-world acute hospital care. And that's how the innovations that I showed you get to patients. That's how it creates the structures, the mechanisms, the processes that stand up for decades and ensure that Irish patients will have access for the best oncologic care for generations to come. And the goal within the EU is to have at least 90% of patients treated on a comprehensive cancer institute over the next 10 years. And hopefully I'll have better to offer than this. And nothing a few stem cells and another 75 years of research can't fix. So I promised I wouldn't keep everyone long today, and I have a few thank yous, but there are just too many people to thank, you know. I've been very fortunate at St. James's Hospital to work with some of the best clinical colleagues that I can imagine. And I mean nursing, medical, our administrative staff. You know, it's been a strange and difficult, few years in the hospital campus, but I'm very proud to be a consultant at St. James's Hospital. I see among my colleagues leadership, I see hard work, patient-focused care, and it's really a privilege to work with people of that calibre. In Trinity, I've just been welcomed, and there are so many people who have made my first few years here just so pleasant, so welcoming, who've been so friendly, who've helped me when I didn't know what to do or how to do things. And I'm just so grateful for that. And I really feel, that the last few years, the welcome and support that I've got, both from the James's campus and the Trinity campus, makes me look forward with optimism and hope to enjoy the rest of my career here with all of you. And it's more important people here. My mum and dad are here. So my dad, Martin, is an engineer by trade, and he ran the State Forestry Company for years, former president of Engineers Ireland. My mum is a teacher of English and history. She actually taught in the inner city of Dublin in the 70s. And that was a time where disadvantaged children really, were not prioritised in terms of education, and my mum would have fought very hard to make sure that those children got a very, very good education at a time when others maybe didn't try so hard. And my parents, they've really given me, I think most importantly, a love of learning. They instilled in all of us a love of education, the importance of education, and very much also the importance of public service and of serving others. And I'm just very grateful for their support, and I'm really happy that they're here today with my two beautiful sisters and my little brother also. Not so little. Yeah. And then there's some other, not this one, some other very important, very, very important people here today. I'll get to you. So James, my husband James, I met James in Teresa's hospital, in Teresa's ward, the Manor Hospital. I was a medical student, James was an intern. James gave me a tutorial on prostate cancer. And it was really very bad. And it was very bad, it was factually incorrect. But he did something right, because we've been together ever since. We have been. And then we've been joined by these two beautiful, beautiful, best, bright and brilliant boys. Lorcan Smith, who's age 8. Lorcan's a New Yorker. He was born in New York, he lived in Brooklyn. He thinks he still has a Brooklyn accent, I'm not so sure. You don't. Cormac Smith is age 4. Cormac is a Dubliner, Northsider, loves football, and Lorcan loves rugby. And I'm just delighted to have them all here. I'm so proud of them. They've really been very patient, very good today, I think, right?
Obviously, they're very proud of you too, aren't you? Words can't express. And what a great ending, Maeve. Thank you so much. Your support team.
Thank you.
Well done.
Thank you.
Thank you, Maeve. And thanks for being so good throughout it all. And James, you did a great job there with the stickers. I was a bit jealous myself, actually. But thanks so much, Maeve, for not just a trip through a very interesting past, but I think also a very optimistic future, and such a nice note, I think, to hit with, like I said, such a difficult disease. And we greatly appreciate everything that you're doing, both for patients and for research and teaching as well, and everything. So we will not have questions at this stage, because I think the audience would be very grateful to get out, maybe talk to you outside. You're all very welcome to join us outside for refreshments. Thanks again. I love your shoes. Very good. I know. Anyway, which I wasn't here. Yes. Because, no, you're okay.